Welcome to this ISTEC Distinguished Lecture. For those of you who are unfamiliar with ISTEC, hello. ISTEC is a CSU-wide organization. It stands for the Information Science and Technology Center. We cover all kinds of areas on campus with respect to the information sciences and technologies. One of the activities that we hold every semester is the ISTEC Distinguished Lecture, which is this particular series right now. Before we introduce our speaker, I will have Sudeep Pasricha from Electrical and Computer Engineering say a few words about our distinguished lecture.
Thank you, Ed. So it gives me great pleasure to welcome Krish Chakrabarty. Krish is just extremely well known in the areas of design for testability, microfluidic biochips, hardware security, neuromorphic computing. He got his bachelor's from IIT Kharagpur in 1990, and the last 3 decades he's spent doing phenomenal work in these areas. He's currently the John Koch Distinguished Professor and chair of the EC department at Duke University. He is a fellow of the ACM, the IEEE, AAAS. In fact, if I start listing all of his awards, we'll be talking for 15, 20 minutes. So I'm going to spare you that ordeal and invite Krish to give us his distinguished lecture.
Thanks for the invitation. I'm very happy to be here for the first time at CSU, and thanks everybody for coming. And today I will talk about our experience over the last 15 years on microfluidics, going from lab research to papers, working with students, commercialization, products, and then ask the question, what do we do next? So that will be the flow in this talk. So microfluidics is basically a bridge. So what you see on this slide is you see some lab instruments. So we basically do all this stuff on a benchtop, and our effort is to miniaturize everything and get it done on a very small chip. So let's get started, and I want all of you to close your eyes. We are trying to look at the future in some sense. We have to dream. You cannot dream with your eyes open, right? Imagine what is possible. You can open your eyes at the end of the seminar, okay, when I'm done. So I want to acknowledge a whole bunch of students, postdocs over the years, faculty collaborators. This is clearly a joint effort, multidisciplinary. I'm from electrical engineering, so I work with biologists and biochemists, biomedical engineers. Our research lab, where we do a lot of chip fabrication, a startup company, my postdoc started this company that was bought by Illumina, and sponsors over the years from various agencies. This is the outline for today. Feel free to ask questions during the talk. Those of you who know me also know that if you don't ask me questions, I will ask you questions, and they are tough questions. So you can ask me questions first. So I'll give some motivation. How did I become interested as an engineer to look at biology or biochemistry? And then look at the technology, microfluidics, and a very specific kind of microfluidics that we use for our research. Some success stories from commercialization, and then look at solutions that we can develop so that we can solve some very interesting scientific problems. And then talk about a gap. So this is a serious problem. There is a gap between microfluidics and microbiology. We are still not able to convince the end users to deploy these chips for their application. So that is a problem, and we are trying to bridge the gap. I will spend the last part of the talk on methods to bridge this gap and then end the presentation. So objective is we want to enable benchtop biomolecular assays for clinical diagnostics, immunoassays, clinical chemistry on programmable microfluidic biochips. These chips can be controlled through software so that we can make them multifunctional, we can make them reconfigurable. So enable realistic biochemistry on simple chips. We don't want the chips to be complex because these would be utilized by nurses and doctors, physicians, not by engineers or computer scientists, but the chemistry needs to be realistic. So doing real chemistry, real applications on very simple chips. So essentially taking all this stuff, all the instruments, all the lab, and miniaturizing everything into a small chip. Of course, we cannot miniaturize the people and put them on the chip, but we want to put their intelligence on the chip. Their experience should go on the chip as well. So there's a paradigm shift in biochemistry over the past many years. So we used to have microwell plates, milliliter volumes, and using test tubes for doing reactions. Skilled technicians, you have to train them and pay them a lot of money to run the reactions, and very bulky equipment. So this clearly wasn't going to scale. Now we have moved to chip scale integration, what is called lab on a chip. The entire lab goes on a small chip. It has reservoirs, it has channels, it has inlets, outlets. We can do all kinds of chemistry on these chips. Yes, please. Good question. So that is the gap that I mentioned. So we haven't been able to convince research labs as much to utilize these chips for everyday reactions. But we have some domain areas, some application areas where these chips are being utilized.
Rather than convincing research labs, convincing educators, because their experiments can be.
Correct. This is a very good point. That is one of the targets we have. So some of the market that we have from the startup company is basically university labs. But the problem is they are not willing to use these chips for everything they do. We think these chips are general purpose. You can use them for everything. They think that these chips are very specific to some small things that they do. So that is the issue right now. So yes, they can use these chips and they are using these chips, but not for all the things that they do. Okay. So let's now go to why biochips. They are small. We can miniaturize them. We can make them this size, so that helps. When you make them small, you can make the volume small. So when you make the volume small, the reaction runs faster, the cost goes down. Automation, you can write software, so you don't need to have as much people in the loop, so less chance for errors. And very high throughput. So this is really the biggest application driver. We can run lots of diagnostic tests in parallel. Right now, if you go to a doctor's office, the doctor would specify a panel of tests for you, and that panel might take 3 days. What we are looking at is running all the tests in parallel, and you can get the results in a matter of a few minutes. So this is the biggest advantage of doing it in a high throughput fashion. Now, what is a biochip? You may not have heard of this term before, so let me walk you through this. So the first thing to note is it is miniature, small, and disposable. This is not like your laptop or your cell phone you will keep for years, but rather you would throw it away after you use it, so it has to be very cheap. And it does a very high throughput screening, or HTS. And what does it do? Well, all the things we learned in college when we learned chemistry. So we did dispensing, we were using pipetting, our thumb used to hurt from all the pipetting we did. We were shaking the test tube for mixing, look for change in color, did a gas come out, was there a pop sound, was there a boom sound, right? And then we can even throw away the sample once we are done, right? So we can do all of these things now automatically on a chip under control, electrical signals or software, a lot cheaper and a lot faster. So this is the reason why biochips are so interesting. Main types of biochips. So the market right now is dominated by microarrays. So these are passive chips. Basically, they have a substrate of plastic or glass or even silicon on which they have patterned some probes using a photolithography process. And then you can introduce a sample, and then you would have some binding based on what the sample contains, and then you can tell what the sample was. The problem with this technology is that most of the work has to be done in advance manually, so you have to bring in prepared samples and reagents and just do the final step on the chip. So we are looking at active lab on chip or micro total analysis systems or micro tasks, where all the active functions, sample prep, mixing, pre-concentration, control of the flow is all done automatically on the chip. So it's a lot more work. It also makes this a lot more exciting to do. So cost is a big reason why we are looking at this. So by decreasing the volumes, going from milliliters to nanoliters or picoliters, we can greatly decrease the cost of doing these reactions. So if you look at the cost of various substances, this is a graph from EPFL, a Swiss university, and you look at copper, which goes into all the interconnects of VLSI chips. It's quite inexpensive, and then silver and gold, platinum cost more, right? And then sapphire, diamond are even more expensive. But compared to this, if you look at the cost of antibodies that we use for doing reactions, they are way at the top. So ampicillin, kanamycin, these are very expensive, even more than diamond or sapphire. So next time your wife, your girlfriend, your spouse asks for a ring, don't buy diamond, buy antibodies. Tell her that antibodies cost more. So this is the ring for you. So let's now come to the motivation. Why do we do microfluidics? Well, if you look at the test tube world, we have no automation. Everything is manual. Nothing is integrated and a very large volumes. You can use robots. There's a market out there where robots can be programmed. The robot arm can go and pick up samples and place them in the places you want, but it's not small. It's bulky, expensive. So with microfluidics, you can make chips or PCBs that are basically the size of a coin, and you get all the three advantages of automation, integration, and miniaturization. And in the realm of microfluidics, we have two main families of chips. The first one is the more mature one, a continuous flow lab on chip, where the idea is that you have a substrate, and then on that substrate, you can etch microchannels. So this is a substrate, and then you have these serpentine channels to enhance mixing, for example, multiplexing, and then you put some sensors to detect the endpoint of the reaction. The problem with such chips is that the channels are permanently etched, so there is no reconfigurable flow. Liquid can only flow along the channels, and you have to integrate micro pumps and micro valves to control the flow, and you cannot make them very small because they leak if you make them very small. And you can use electrical methods, electrokinetics, electroosmosis to drive liquid, but then the voltages are very high. There are thousands of volts you need to drive those chips. So we were looking at something different where we have a pattern array of electrodes. We can control them electrically. We can apply voltages from outside, high and low. So think of this as ones and zeros. And we can trap a droplet at a specific location, or we can move them around as we want. We can do mixing and dispensing and all kinds of fun stuff. And we can also integrate sensors on these chips. So we call this digital because a droplet is like a bit of information. This is my payload. It can carry DNA, can carry protein. That's my bit, and I have a clock signal, and that can basically control the voltages that I apply to each of the electrodes. So let's now understand the science of why this works. So this comes into play at very small volumes. When the surface to volume ratio is very large, then the bulk forces get very weak and the surface forces get much stronger. So imagine you have a surface here, an electrode, and then you coat that with an insulator and a very thin hydrophobic layer that I have not shown in this picture. Then because the surface is hydrophobic and the droplet is in the range of nanoliters, the bulk forces are weaker, so I get a nice sphere. If I now apply electric field between the electrode and the droplet, then this electric field would basically change the surface from being hydrophobic to hydrophilic. The surface energies get affected, and the droplet wets the surface, and the contact angle will change. And this effect is fully reversible. I will show you a video here of a lab experiment. This is the droplets on the chip. And when I start the video, you will see there is a period which is quiet, and then we'll get a pulse, and there'll be one movement, and then I increase the frequency of actuation, and you will see the droplet pulsing more often. So that was the first one, and now you do it more, so up and down. So this is how it works. Okay? So the original science of electrowetting has been known since 1870, the first paper in French. I had to read that paper in French to understand some of the science of that. Now, fortunately, I had some French in school, so I could make some sense of that paper. Now let's look at the physics a little bit more. So essentially, you have the solid layer, and then you have air and liquid. So you have the various surface tensions between the interfaces, solid gas, liquid gas, and solid liquid, and there's a contact angle theta shown here. So typically, these two remain constant, but when I apply electric field, the solid liquid surface tension would change because I'm adding extra energy into my system. Think of this as a parallel plate capacitor, so the energy has to go somewhere, and that goes into changing the contact angle. So now you can go from the up-down movement that you saw in the earlier video to movement on an array. And to do that, I can go from this picture, which is one electrode with a droplet, to having multiple electrodes with a small gap in between and a top plate, which is permanently kept at 0. I permanently ground that plate. If I want to hold a droplet at this location, I would apply a high voltage here. So high, low, low, and the droplet stays there. To move it to the right, I would make this low and make this high, keep this low. That puts a net force that drags it to the right, and vice versa to the left. Okay? And I will need some oil to fill the gap between the two plates, because this is a very small volume, nanoliter, picoliter, and I've seen experiments myself where after about 5 minutes, the droplet is gone. It's evaporated. Poof, where's my droplet? Poof, it's gone. Right? So we use oil to prevent evaporation. Also, cross-contamination is a problem. If I'm moving large molecules like DNA, protein, they would stick to the surface, or biofouling can happen, so I would use oil to minimize the chances of that. I saw somebody raise a hand. Yes? This is an excellent point. So the first thing to note is that there is no current flowing through the droplet. The current in the vertical field is 0 through the droplet. That's number one. Number two, the way this works is a surface phenomenon. So an electrical double layer gets formed at the interface. So you have pluses and minuses forming on the interface, but it doesn't occur in the bulk. So there's no change happening to the droplet itself. And note also that this would work only with liquids that are polarizable, where you can have pluses, minuses. So if you want to move benzene, petroleum, it doesn't work because those are not polarizable liquids. But if you can move salts or water, this will work. Okay? The oil is not to make it polarizable. It's already polarizable. The oil is, as I said, to prevent evaporation and to prevent cross-contamination. That's correct. I don't want to change the oil. I want to move the droplet that's encased in the oil. Okay? I could spend a whole day talking about the physics and the chemistry. I had to learn that. I wasn't trained in this. When I was in college, I didn't have classes on this. This is all stuff I've learned. It just shows you that learning never ends. Okay? You need to keep learning new things as you go along. So for those of you who work with algorithms, this is like a finite state machine, right? Think about the electrode array as being a vector of ones and zeros. High voltage is a 1, low voltage is a 0. And you have a current state, which is a vector of ones and zeros. Anytime a droplet moves, you're doing a state transition. So you can formulate some very interesting problems in routability. Can you read certain states, verification, and so on. Okay? I'm going to show you a video of a droplet movement from the side. So you will see the droplet marching left and right, left, right, and so on. Let me show you one more video from the top. So this is a chip we had fabricated about 10 years ago. And you will see that a droplet is being routed very fast. This is the real speed, not simulation, to the east, north, west, and south. So this is how the droplet transport happens. Okay? I would like to ask you a question. I was expecting a question, so you didn't ask me, so I'll ask you. Why is it that the electrodes are shaped in the zigzag fashion? Why isn't it a straight line? In the earlier videos, earlier pictures, you saw just a rectangle, right? So what is it here? Why is it that I have this zigzag? Can you explain that? Yeah, go ahead. Surface area, very good answer, because we want to pull the droplet to the right, it must have an overlap with the next electrode. That's how you can pull it. This is a nice intuitive answer. From the physics point of view, you have electrical lines of force, you want to concentrate them. If you have zigzag, you get a very concentrated lines of force, so you can pull the droplet. But I have an answer that is more intuitive. If I want to pick this guy up from here, and I use only my palm, I can't do that. It's very hard to lift it up, right? My palm cannot grab it, but if I use my fingers, I can pick it up. So zigzag means you have the fingers now to pull the droplet to the right. You get lots more videos if you go to the website on various operations. Let me show you an operation of split and merge. So we can think of this as a microfluidic instruction set where the instructions are not add, subtract, multiply, but rather dispense, merge, split, move, heat, and so on. So you have two droplets of volume 1x, and they are being brought together. You form 2x, and then you do a split, and you get back the original droplet. Two more videos. The first one is droplet formation. The outside world is not digital. So you see when you look at embedded systems, you have A to Ds, right? You get analog signals, and you have A to Ds, and you get your binary signals after that. Similarly, we have reservoir, which has bulk volume, and then we want to generate droplets that can be fed into the system. So this is a line of electrodes. I can control the voltages here, and by that I would create a neck, and I can break the neck. So you will see here what's happening. I control the voltages, I form the neck, and then I break the neck, and I can inject the droplet inside my system. This is my A to D operation. And the next one is a dance. I have many droplets. I'm going to choreograph them. They will all move all together, and because they are controlled by software, they don't accidentally hit or collide. I know exactly where they're going. So this throws up the option of parallel processing or multitasking, pipelining. So we can run multiple reactions at the same time because we can control multiple droplets on the chip at the same time. So we were able to find a lot of good applications of this technology in molecular biology. So DNA amplification, polymerase chain reaction, DNA pyrosequencing, immunoassay, protein-based, and MALDI. For example, here, cell culture, we can also culture colonies of cells on these chips. We can feed the cells using microfluidics. And cell lysis, we can open up the cell wall to extract the nucleic acids from the cell. Electroporation, we can bombard the cell and we can inject things into the cell. So all these were very nice. We got a lot of applications of microfluidics. Then what happened is we looked at commercialization. And there was a company formed out of Duke called Advanced Liquid Logic, and they had a product in 2011. This was the first product. So you basically see a PCB product where you had reservoirs on the edges for loading reagents and samples. And there was a region where you could transport the droplets, incubation region, and then some optical region to sense. This was some of the specs of this product. It was not very small because it was PCB, so you couldn't scale it down too much. And also we managed the cost by not fabricating every electrode. So we only fabricated electrodes where we had a need. This is how it looked. So the cartridge was basically throwaway, was placed right here. The instrument was pretty big. It had all the power supplies. The software was loaded, and the user could see things going on here. Okay? So at the time this product was announced, there were 2 counties in Illinois that had mandated that every newborn had to be screened with these chips. So we were selling 500 chips a day at that time, with 2 counties. Of course, the margin was small, so it wasn't making that much money. But imagine if you had more counties or more states, the market could be huge, right? So the promise was there, so there was an acquisition. So Illumina, which is the world's largest DNA sequencing company in San Diego, they bought Liquid Logic in 2013. And their senior VP basically said at the time of acquisition, we would like to use this for our next generation sequencing workflow. And what that means is sample prep. So a lot of the time goes into sample preparation. You need to prepare a whole range of concentrations, 1 molar, 1.2 molar, different volumes, so that you can run your various diagnostics. So he made this announcement, and we thought it will take 5 years for this to be a product. It usually takes time for a startup to go to a product. But we were very happy at the time because if you look at the 50 smartest companies from MIT Tech Review, in 2015, Illumina was number 3. So it's good to be bought by the 3rd smartest company in the world, right? And be in the same league as Tesla Motors or Xiaomi, Alibaba, and all these companies, right? All right, so then within a year, they announced the product. So this was called NeoPrep, Library Prep System, and basically it was used for generating all the samples that are required for doing sequencing. So what I'm going to do is I will play a movie. I need to get my volume up. Okay, I think I blank this out. So max my volume. This is not the volume. Sorry, I'm trying to find my volume here. Okay, this is maxed out. And then what I'll do is I'll play a movie from Illumina that shows a scientist, and she is very busy doing everything by hand. She's doing, using the shaker and centrifuge and preparing stuff, and she's getting very tired. And then she will see light, and you will see the chips come into play, the music will change, and you'll see the ad from the company. It's tedious. So she's exhausted. And then she will look up and she will see light. And now you see the music change. So now the droplet, one droplet, and now you see many droplets. So they were able to utilize hundreds of droplets in parallel. Each droplet consumes only 1 microwatt of power. So even with hundreds of droplets, it's still with very low power. And she has a smile on her face now, a smirk or a smile. It'd be a smirk. Okay, all right. So let me go on. So then we also were able to find one more company that licensed this research, Genmark, and they have a system called ePlex, which gives you very rapid flow for nucleic acid extraction, amplification, and detection. So less than 2 minutes of hands-on time, and they can get from sample to result in about an hour to 1.5 hours. So I will play a video of what they do.
Genmark is pleased to present the future of molecular diagnostics. ePlex, a sample-to-answer multiplex molecular diagnostic system, delivers laboratory efficiency and rapid comprehensive clinical results for clinicians and patients. ePlex features enhanced workflow, enabling optimal use of staff time. Simply load the patient sample into the cartridge, scan the cartridge barcodes for positive sample identification, and load the cartridge into any bay. The visually appealing user interface is intuitively designed to meet the unique needs of laboratory professionals. The fully integrated cartridge contains all required reagents.
So the droplets are being injected now into the system. And you can see they're moving really fast. This is faster than what we could do in the lab. So you can see there's always an advantage of commercializing.
Automates molecular testing, including sample preparation, extraction, amplification, and detection to deliver rapid sample-to-answer results.
Okay, so I think I can skip the rest of this video and instead go to a third company called Babies, which is also licensing this research.
It's a high-throughput platform that quantitatively measures the activity of lysosomal enzymes in newborn dried blood spot specimens. Reduced activity of these enzymes may be indicative of mucopolysaccharidosis type 1, Pompe, Gaucher, or Fabry disease. Everything your lab needs for LFT screening is in one small workstation. Seeker is made up of 4 instruments, one desktop PC with pre-installed SpotLogic software, backup power supply and accessories, all fitting on a standard size lab bench. Seeker integrates easily into your lab workflow. On 2 workstations, Seeker can screen up to 960 samples per day.
That's a lot.
For more request a demo, please visit.
So you can see that we were able to go from basic research in the lab and work with students, postdocs, then commercialization and products. So one of the downsides is that now these companies don't want to talk to me. These were my people, but now they tell me that I go and give talks and tell the whole world what they do. So I'm persona non grata inside their organizations. That's okay. That's part of big research. I'm very happy to see that they are making progress. So now let me talk about some of the other things we did, which these companies are also using, and that is of interest to the EDA world. So can we now talk about solving interesting design problems? So if you think about how do we map a protocol onto the chip for automated execution, we take the samples. So we have, for example, plasma, serum, urine, saliva, say 4 kind of samples, and we want to do 4 assays. We want to figure out the concentration of glucose or lactate, pyruvate, glutamate. We could do them one by one, but that's very slow. So can we interleave them? Can we find a way to run these tests in parallel? So we can follow the same route that was followed in VLSI design or microprocessor design, ASIC design, where we start from a description of the application, and then we design the architecture. So how many blocks of each type we need, at what time they run, what kind of scheduling we do, so scheduling and binding operations. And then we look at the actual layout, the geometry of the chip, where do we place the different modules, and how do we route the different droplets, and then we get the actual chip. So we basically can think about two kinds of optimization problems here. One is at the fluidic level, we have to do the scheduling. We can build a graph that models what kind of protocol this chip will run. And then we can talk about scheduling when we run each operation in points in time. And then we can talk about placement of the modules and routing of the droplets. So all this is done for the actual fluidics. Now we have to do the chip design. So we have to now think about, at the chip level, which electrodes are being utilized at which points in time. And then we think about the addressing problem. Where do we put the control pins? We put them underneath the microfluidic layer. So we have the electrical signals that go from below. And then we have to think of the wiring. How do we connect the pins to the various electrodes? We have tools for doing that automatically. So the user would say, this is my protocol. So we can create a model like this for the protocol. We have a library that can be characterized experimentally in the lab. We can run experiments to see how much time each module takes and what is the area of each module. And then we have a spec in terms of how big a chip we can have and how much time this experiment should take, how many reservoirs, how many sensors. And then the output of this through the tool would be the resource binding. So which operation gets mapped to which resource, scheduling, at what point in time each operation runs, placement, where on the chip we place all the modules. And we want to ensure that it would finish under the specified time, and also the area is under the maximum specified area. Question?
Are these the sort of problems that are now being solved internally by your commercial?
Yes, so these are following the same path that was followed in VLSI design, but you have to take into account the domain-specific things, because if you look at electronics, you have an unlimited number of electrons and holes. Fluidic samples are of limited volume, so you have to worry about how many droplets you can get. So there are a lot of such constraints, and also about spacing. So when we talk about routing droplets side by side, we can't bring them too close because they may overlap and accidentally mix. There's a lot of inertia that comes into play.
My question was more about, are these problems academic research, or are these the problems that Illumina is going to solve?
These are academic research problems. These have led to papers. So Illumina, they don't have the applications that can utilize these tools. So we are working on helping them identify applications which can use these tools. But these tools are still not being used in the actual chips out there in the field. The chips are that you saw, they only do extraction, routing, transportation, and some mixing. That's it. But they don't do this level of detailed automation. Yes, question. Can you please speak up? I cannot hear you. Yes, so we have an operating system and a compiler. So the compiler is what generates the plan of moving the droplets. And we have an operating system with some built-in sensors, so we can track either using CCD cameras, or we have capacitive sensors under the electrodes, so we know where the droplets are. That's correct. Yeah.
So you're talking about using
Yes.
Would the every material would respond differently to
That is a very good point. So the question is, when we have this electrode and then we put on that a hydrophobic layer or insulator, what effect the type of material has. So we have experts who analyze that. So this is an interdisciplinary project. We have a big team. So there are people who look at what is the interaction between this liquid and this material. So right now the coating that we are using is a mixture. It's actually tantalum perylene and tantalum pentoxide. Now don't ask me why, because I'm not the expert, but that's what seems to work, right? So there's a very interesting question about the actuation voltage versus degradation, the breakdown voltage. So if you actuate at a very low voltage, the droplet won't move. If you increase the voltage, the droplet moves faster, but then at some point you reach the stage where the insulator breaks down. So you need to have the right balance between the breakdown voltage and the actuation voltage. Yes. Very sensitive. So it can be in a truck while you are driving the truck. You can run assays in the truck. We haven't had issues about background vibration. So it's not that you need to have an optical bench to run experiments. So we run this on a normal bench You can have people walking by and a cart going by, it doesn't matter, but you can have the surface moving. All right, so then we look at another interesting problem of how do you place the module. So once you have done the scheduling, you need to figure out where to put the modules. Now this is similar to VLSI placement. You have a X, Y coordinates as shown here, and you would like to place the module here. But the big difference from chip design or microprocessor is that your modules are virtual. They come and go. So unlike in a processor where if you put an adder here, you cannot put a multiplier there. You have to put it somewhere else. But here, every module has a lifetime, and once the lifetime is over, you can put something else there. So this is like 3D bin packing, but it's a computationally expensive problem. So if you have scheduling done before, you can transform this to a sequence of 2D bin packing, or 2D packing problems that is much simpler. So let's look at examples. This is a very common benchtop protocol for protein assay. You have a high concentration protein, you want to dilute it down. And the dilution is done through an exponential algorithm. So you take a droplet of protein, mix it with 1 droplet of water, and then you do a split. So you get C by 2, then you get C by 4. So this is how it goes. It's a binary algorithm, and it's very, very tiring to do that by hand. So we want to do it on the chip. So we took some specs, and then we did a chip design. So this is how the 3D bin looks. So every entity inside this bin is a microfluidic module, tells you where it is placed and when it is activated. That's the time axis, the z-axis. And then the area of the chip was 9 by 9, which is smaller than the maximum 10 by 10, and the optical sites are also indicated here. If you have certain electrodes that are faulty, if you can test your chip and those electrodes fail, you can still make money on the chip. You can still do the reaction on the chip because you can bypass those faulty locations using the software. So if I know that these electrodes are bad, I would draw these vertical black towers. So I wouldn't put anything there. I would put modules around it. So the impact is that my assay time would go up a little bit, 6%, but I can still make money on the chip. So this is a lot like how Intel sells microprocessor chips. If you are buying a laptop and you customize it yourself, you have an option of you have processor 2.3 gig or 2.4 gig or 1.8 gig, right? And the price point is different. You think that these are different chips? Intel is fooling you. Don't tell Intel, by the way, I'm on the camera, right? So what happens is Intel has the same architecture, the same process, and they get the chips back. Then they do speed binning on the chips. They see at what speed the chips are failing, and they put them in bins. And then they would price it based on the bin. The faster bins get them more money. So we could do speed binning or some kind of binning even here. We could sell these chips at a lower price because the assay time is longer. And then there's also the routing problem. So droplet routing is again similar to VLSI routing because it's a matter of minimizing the time it takes to route. But it's not the same problem because the routes are virtual. So you don't need a permanent channel. So in VLSI routing, you need a wire shown in red. In droplet routing, you can just route the droplet, then you can use the electrodes for doing something else. Okay, so a lot like FPGA design. So this is like train tracks. So if you think about how trains are routed, the tracks can cross. That's no problem. But the trains must not come at the same time. Otherwise, there's kaboom, right? So you can use control algorithms to make sure the trains are staggered. Similarly, we can control the droplets when they show up at the intersections. And then there is a problem of the number of pins. So initially, I told you we can control every electrode individually, and that was a lie because it's not possible. Because if you have 10,000 electrodes, you cannot have 10,000 pins. If you look at these chips, I'm sorry, I don't have a chip with me now, but the pad frames are very large. So the boundary of the chip is big circles, these are the pad frames, and the inside is very small square, the microfluidic part, right? So the problem is, can I control a large number of electrodes with a small number of pins? Now, in one hand, I can do that if I move only one droplet at a time. If I do it serially, then of course I can do things with few pins, but the challenge is, can I do things in parallel? Can I still have high throughput and use a small number of pins? So this could be done by using an idea called cross-referencing. The same way we address SRAM and DRAM memories, we have rows and column addresses. So we go from n by n times m to n plus m, n rows and m columns. So this way we have fewer number of pins, right? So this we can do, but there is a problem that comes up in this domain that is called electrode interference. And I'm going to explain to you what the problem is. So this is an example of a 10 by 10 chip, 10 rows and 10 columns. And I have droplets 1, 2, and 3 shown here. And I would like to move droplets 1 and 2, so I activate the columns and I activate the rows so that I can move these droplets, right? But when I do that, I have my destination cells, and I also have 2 unintentional destination cells, right? So now when I move droplet 1 and 2, 3 also moves. That is unintended. That's a problem. So for this case, I cannot move 1 and 2 at the same time. So now if I scale this up and I have 9 droplets and their intended motion is shown with these arrows, the question is, how many steps do I need to move these droplets? One extreme is 9 steps. I can do 1 by 1, 9. It's doable, but very slow. So can I do anything better than that? And the answer is I can do that by forming a group, right? So I basically make an observation that if 2 droplets have a common destination row or column, then I can move them at the same time. And I use that observation to form a group of droplets that can be moved simultaneously. And I do that by building a graph. So every electrode or every droplet gets mapped to a node in the graph. And I have an edge between 2 nodes if the corresponding droplets have a common destination row or column. That's the rule I use to build the graph, right? And once I build the graph, I do a click partitioning on the graph. The number of clicks on the graph tells me how many steps I need, and all the droplets in the same click can be moved at the same time. Okay, so this way I can find out how many droplets I can move. So you can see you can go from chemistry and biology to algorithms and math, right? You can make this transition very easily. There's also another interesting thing you can do with routing is, note that it's not just ones and zeros. There's also a don't care state. So if I think of a droplet which is located here, and I want to keep it here, this has to be 1, these 2 have to be 0, but this one can be anything. I don't care. So it's an X, right? I can map it to 1, I can map it to 0. So think about a movement that I want to do like this from E2. So I can draw this table which shows the actuation over time. This is the first clock cycle, electrodes 1 through 8, second clock cycle, third clock cycle. So in every clock cycle, I have many electrodes that are X's, right? So now the question is, can I map the X's intelligently to 1 and 0 so that I can make some columns identical? If some columns become identical, then I can broadcast. I can have the same control pin drive these columns at the same time, right? That's why it's called broadcast addressing. How do I do that? I once again build a graph where every electrode is a node in the graph, and I draw an edge if they are compatible. Now what is compatible? If in any clock cycle they don't have specified 1s and 0s, then they're compatible. So for example, in this table, 1 and 2 are not compatible because they have 0 and 1 in this cycle. But 1 and 3, but 1 and 4, for example, if I look at 1 and 4, in every cycle, either they have the same value or it's an X value. Then I do a click partitioning on this graph. The number of clicks tells me how many pins I need, and all the nodes in the same click can be controlled by the same pin. Okay, so this is very useful, and this can be used to make the chips simpler. All right, so now I come to the next part of the talk, which is all this is great, right? We have a thing that works very well. We have commercialized it. There are products. We also have nice algorithms. But there is a gap that bothers me. Okay? And let me explain to you the gap. This is tracking the number of papers that people publish with microfluidics in their keywords or the title or the abstract, right? So the blue graph is showing the number of papers in engineering journals, the kind of journals where I publish my work, right? It's growing super fast. That's great. In blue and in green and in brown are the number of papers in medical journals or interdisciplinary journals. It's growing, but it's going very slowly. And we have here basically a gap, right? Oops, sorry, let me go back here. This is the gap. So this gap is a problem, right? Let me see if I can play this small audio clip.
Mind the gap.
Do you know what this is? You don't know what this is? Play once again. Who can tell me where this is from, this clip?
Mind the gap.
You know this? No, not Denver. London Tube. It's a tube in London, exactly. So thank you. That's the. So why do we have this gap? I have tried to think very hard and deep about this, because if I go to NIH and ask for funding for the research, this is the graph they're going to show me and tell me your research is useless. So all the work I've been doing is not really making an impact. Yes, Sanjay?
This y-axis, is it just linear scale or is it a log scale?
We can tell from the number. I think, I'm not sure.
You're trying to show an exponential growth on the y-axis.
That is true, but I don't know if it's exponential. Maybe I shouldn't say exponential. If I said that, I take it back. It's a rapid growth, right? And this is a slow growth, and the gap is growing, right? So this is not a good sign, and the reason why we have this. Okay, thank you. I should, because then this graph would probably be flat, right?
Yes.
Yeah, okay, true. So why do we have this gap? There's a gap of trust. So when I talk to people at Duke in the medical school, doctors, they tell me that we have a trust issue. We don't trust your devices. Why? Because we still cannot support quantitative analysis. We can detect presence or absence of things, but we can't tell very clearly how much, right? And that is necessary for gene expression analysis or for immunoprecipitation reactions. So that is a problem. Second is real-time sensing. There was a question from the back, I think from you, it's a very good question about how do we track the droplets. It's hard. We cannot do it very well. So real-time sensing isn't enough. We need to do much better real-time sensing. And third is defects. The chips, these chips are pretty bad. You know, when we fabricate them in our foundry, we get one chip that works on a wafer, right? Maybe two if you're lucky. And after 10 minutes, the chip dies. So there are problems of reliability and yield. So how do we improve that? So this is my new research. I'm now looking at these as the research topics. So one of them is, can we look at biomolecular quantitative analysis, or BQA? These are far more advanced protocols. Now it's no longer just showing droplets can dance around. I can make lots of videos on that, but it's not going to help me, right? I need to show that people can run such reactions on these chips. So if you think about what they do is they would first get your samples, do cell culturing, we can do it on chip, and then they would assess the quality of those chips. We can also do it on chip, we can sense, and then we can do cell lysis, we can open them on chip, but it's not a path which is a straight line. There are loops in this protocol. So we may have incomplete analysis, we may have impure samples, we may have to go back and do things once again, and it's very hard to predict how many times this will run, okay? So what we don't know is the connectivity between these different points. So we know what the points are, but we don't know how they are linked to each other. So we need to create some modeling for that. So to bridge the gap, we basically need to create support for independent and multiple sample pathways, support quantification, and also model the uncertainty about the sequence that the pathway will take. So this is the kind of modeling we have to do. So each of this is a graph you saw before. This models the actual protocol, and these are okay as is. So basically the sequence of steps inside each subgraph is known, but what we do not know is in what order we execute these graphs. So that's why we need a control flow, right? So this has to be modeled as a control flow, so we can have mechanisms to synthesize arbitrary execution orders of these graphs. And then we have defects. I'm going to show you a video of a chip which had a defect. I wanted to mix or merge these two droplets, okay? And what you will see is a bomb explosion, okay? So it's not a bomb in the room, it's a bomb inside here, right? So, okay. So the two droplets are being brought together. This one explodes. We try to still keep going, try to forcibly merge them, and they explode. So what happened is electrolysis. So under the droplet was the insulator that got broken up into fragmented islands, and that caused droplet electrolysis, and the droplet just broke. So this is one example of a defect, okay? There are many other things that go wrong. So this is a graph showing how much voltage you need for droplet actuation. So in the early days, it's very good. It's pretty flat curve. And then you start seeing a linear increase in the actuation voltage. You can still use a chip here, but beyond this point, it grows. This is exponential. We have measured this exponential. And then the chip cannot be used. So this is a failure of the chips. The lifetime is fairly short. And then dielectric breakdown can happen. Sometimes physical particles, the electric field gets concentrated. So we have some pinholes and so on. And then there's residual charge. So in electrowetting, ideally, the charge should go away. When you have the electrical double layer, you take the voltage away, the charges should spread out. But they don't always do that. So you can have charge accumulation under the electrode. And essentially, the forces are now not equal when you do a split, and you get unequal volumes, as you can see in this picture. So we did experiments to see, can we use software to mask these problems? So we fabricated chips, some details. Let me not go into the details. but we wrote some protocol that we mapped onto the chip, basically a mix, split, keep a spare droplet, take one droplet, do another mix, do a split, and you keep a spare droplet. So we had some other stuff we did. So this is the chip we later. I told you that we have a very small fluidic part, and the I/O pad frame is pretty big here. So let me skip this. This is a movie I want to show you. So just a quick walk through the video. This is when there was no fault. So the checkpoint, there's a sensor at this corner. So we do the mix operation, and then we do a split. We try to find the droplet. We found it, so that's good. And then we do the mix number 2, like this, and then we do a split, and everything is working fine. Then we had a chip which had a fault in it, and we didn't know where was the fault, right? So we again had the checkpoint shown in red. We did the mix operation, and then the droplet could not be found. So do the mixing, splitting, droplet could not be found. We don't know where is the fault, so we use the alternate route. We take the spare droplet, and we route the spare droplet using some other route to bypass all those locations, and then we use it for the second mix operation, and then everything works fine after that. So we gave this chip to a user, and the user could not find any problem. They told us this is a very good chip. Thanks very much. So that is the point. Can we use software to basically bypass all these faults? This is a new product from TSMC. So we work with a Taiwan University and TSMC to go to a real foundry. We don't make the chips anymore in the lab. We make it in the foundry. A very old process, but it works, 0.35 microns. So now we have microelectrodes, lots of them, which have transistors underneath them, so we can detect and sense every microelectrode. This is showing you a diagonal movement, which we can do now, which was not possible earlier. So chips droplets can not only move XY, can also move in a diagonal fashion. We can also manipulate droplets of 2 different sizes, small and big, so we can bring them close like this. And then we have the next problem is the fouling. So if you think about this here, we have 2 droplets being routed. The first droplet leaves behind some residue, and that contaminates droplet number 2. So instead of that, we could now use an alternate path. So the second droplet has no overlap with the first droplet, but the time goes up, so that's not good. We can use a wash droplet. So now we have basically a wash operation. The first droplet leaves behind residue. We bring the second droplet, and it waits until a wash droplet comes and cleans up the mess, and the second droplet can pass. The problem is the second droplet had to wait. So instead, we could be more intelligent. We could interleave. So we get the first droplet in, and then we send the wash, and the same time. So by the time the second droplet comes, the washing is done, right? The problem here is that this video is not real. The wash droplet has to be very large. If I have a spill on the floor, I need a mop that is larger than the spill. So the wash droplet has to be 2x or 4x, and it moves like very slowly. It doesn't move like this that you saw in the animation. So Udeep is looking at me. I need to finish up. So we have some new project on acoustofluidics. We're using sound waves to move droplets. And the advantage is that there is no contact between the surface and the droplet. It swims in a film of oil. The droplet literally swims. So with that, we can avoid any cross-contamination. Let me skip these slides. So we have a preliminary hardware design. It's not yet that nice, but we are just trying to show that this works. We can use acoustic waves to move droplets, and some videos that show that we can do mixing using acoustic waves, and we can also route droplets as shown in this picture. So with that, let me go to conclusions. So basically, I walked you through lab on chip and microfluidics, how we can use software to control operations. We have commercialized this product, but there's still a gap, and the further growth is stymied by various limitations. But we have promising paths forward thanks to MEDA and acoustofluidics. And the numbers, if you're interested, these are the numbers for the smallest volumes and the smallest voltages and the smallest electrodes. So 7 volts is the smallest volume for routing, 11 volts for dispensing materials and electrode size, and so on. So we have a biochip ecosystem. We have on one side the people who build these chips and the users, and then we have the software, which is the bridge between these 2 chips. So with that, I finish my lecture. I have something else I was hoping to walk through if you give me another 3-4 minutes, but it's up to you. I mean, it's not about this topic, but about something else. What do you think? Can we do that? I promise it will be fun.
Okay. If it's fun, sure.
Ed says no. You say no?
Well, I mean, it's not the read, but okay, go ahead.
Okay, let's go ahead.
Spending more time saying no.
Okay, so because many of you are young, you are students or trying to build a career, I want to walk you through some stories, right? So this is actually from Jürgen Becker, my colleague in Germany. So this is translated from German, so some of it got lost in translation. But here are 3 lessons to help you succeed. First lesson is, a crow was sitting on a tree doing nothing all day. Okay, this crow is lazy. A small rabbit saw the crow and asked him, "Can I sit like you and do nothing all day?" And the crow said, "Yeah, why not? You can also be like me." The rabbit can't climb the tops at the bottom of the tree. The rabbit sat on the ground below the crow and rested. A fox appeared, saw the rabbit, ate the rabbit. Okay? End of the story. So what's the moral of the story? To be sitting and doing nothing all day, you must be sitting very, very high up. Okay? Then you will be safe. That's the moral. Second story. A turkey was chatting with a bull. That's my turkey, that's a bull. I would love to be able to get to the top of the tree beside the turkey, but I haven't got the energy. Okay? Well, why don't you nibble on some of my droppings? They are full of energy. You'll get nutrients, right? So that's what the turkey did. He picked at a lump of dung and found that it gave him energy. So he started getting stronger. So he's getting stronger and stronger, had more dung, and he reached the top of the tree. And then what happened is a farmer came, spotted the turkey, and shot the turkey out of the tree. And end of the story. So what is the moral of this story? The moral of the story is, bullshit might get you to the top, but it won't keep you there. So you have to be genuine, you have to be real, okay? You can't get to the top by bullshitting. The last story, the third story. A little bird was flying south to the winter, for the winters, that's my bird, and it was very cold, so the bird froze and fell to the ground. This is the bird on the ground. And while it was lying there, a cow came by and dropped some dung on it, okay? And the dung is warm, so it started thawing the bird. You know, the bird started waking up, you know? And he lay there all warm and happy and soon began to sing for joy. That's my bird singing, okay? And then a passing cat heard the bird singing and came by to investigate. And then he found the cat, he dug him out and ate him. End of the story. So this story has multiple morals, okay? The first moral is, not everyone who drops shit on you is your enemy. The cow was not the enemy of the bird. So if your advisor gets upset with you, he's not your enemy. Not everyone who gets you out of shit is your friend. So the cat was not the friend of the bird. And when you're in deep shit, keep your mouth shut. Okay. Thank you very much. Okay, that's the end of the talk. Thank you, Ed.
So we don't have time for questions, but I just want to quickly announce that Krish is giving a second talk today from 3 to 4 at NSC 308. It's going to be about 2.5D 3D chips and the future of those sort of chips. So please attend if you can.
Thank you.
So we're just going to end by presenting to the distinguished lecturer a plaque. So let me read from it. Presented to Krishnendu Chakrabarti in appreciation for your Information Science and Technology Center distinguished lecturer.
Thank you very much.
Thank you all.